have you by any chance come across these promyelocytic cells? And of course, some of them look like angel wings. And you could also find promyelocytes that have got granules within them, and these are azurophilic granules. And lastly, you can also see blast cells in acute promyelocytic leukemia. Since you know this already, do we know the genesis of that? And do we have any idea how the cell cycle relates to this type of leukemia? I will spend this short time for us to go through and have a lovely time understanding how the cell cycle processes relate this to how Apml comes out of this. We should just note that it is not the cell cycle processes that leads into Apml. But remember, central dogma theory says that before a protein synthesis, there is either an RNA or DNA polymerase transcription of certain information on the DNA. And that brings about the messenger RNA that has the correct base pair for protein synthesis. And in the process of this transcription, there are different genetic aberrations that could happen, and to list just a few, we know of translocation, we know of deletion, and other mutations. One of these is called the translocation of chromosome 15 and chromosome 17, producing an oncogen, and this oncogen is what leads into Apml. We will just delve into that and understand the exact process that happened, and if the oncogen is produced, how exactly is it going to elicit its effect in the genetic environment to lead into this Apml. Now, onto your basic understanding of the cell cycle. We know the cell cycle has got very interesting stages that go, and each stage has specific processes that happen within it relevant for the cell that is undergoing mitosis. You have the G0 phase, and the G0 phase is called the resting phase. At that phase, the cell has left the cycle and it's not dividing, but it is undergoing a metabolic activity. So it just experienced the metabolic activities, but it is neither preparing or being ready for mitosis. But the actual cell cycle is called the interphase, and in interphase has got two big G's, and inside them is an S. So we have a G1, a G2, and an S in the middle. The G1 is the stage where the cell begins to prepare. And in G1 phase, what happens there? Remember, a cell that is preparing to undergo a division, it must duplicate its cytoplasmic organelles. So you can't say we are going to divide and have one cell having mitochondria and the other lacking it. One cell having an endoplasmic reticulum and the other having it. So you need to duplicate them. So in G1 phase, duplication of organelles happen. What again? When we duplicate and produce a lot of protein, the cell will become tensed. Then it has to enlarge so that it can accommodate all these proteins and organelles that are duplicating. So in G1 phase, remember, the cell enlarges. And to move forward with that, remember what we have just talked about is only regarding the cytoplasm. Then the synthesis stage will now be related to the nuclear environment. So inside the nucleus, we've mentioned it several times that there is chromosome, and that chromosome is not the crystallized X shape that we know as chromosome, but rather this is chromatin lines. So there are strands of chromatin lines within this nucleus, and that duplicates. This is what we call synthesis stage. So the S stage stands for synthesis. And the last G, which is the G2 stage, is where we look back onto all the processes that have gone before. That is the G1 phase and the S phase, to see if there were any errors that happened within there. We have to correct them, even though there will be more growth occurring within the G2 phase. But the relevant aspect is the checkpoint, where there are repair of errors if there is any DNA breakage or if there is any aberration that have occurred there. Don't forget our discussion on the chronic lymphocytic leukemia, that in these stages there are checkpoints where the cells make sure, or the body makes sure, that it is not transferring any error into the cell division. So we finished the interphase that has the G1, the S, and the G2. The next stage of the cell cycle is the mitosis, and in that stage the cell is ready to divide. So in an overview understanding of the cell cycle, it is a preparation for eukaryotic cells, and I mean cells that have membrane-bound nucleus, to duplicate themselves. So for instance, you go to gym and you want to build your muscles, so you take a lot of proteins in there. Is it the protein that is going to accumulate over there? No, but rather there is transcription of information called messenger RNA within there. That gives the cell an idea that we want to build muscles, so that the tRNA will go around your body and gather amino acid molecules, and these are the basic elements regarding protein synthesis. So in the cell cycle is the preparatory stage for cell division, where mitosis occurs, and of course we can sing a song with mitosis that we know that mitosis have the PP mat, that is the prophase, prometaphase, metaphase, anaphase, telophase, and when these processes are finished, it means the cell is done dividing. So in the phases of this cell cycle, and just like I mentioned the checkpoint, there is a specific enzyme that is to see to it that there is a correct cell cycle progression, there is DNA replication, and there is phosphorylation of the gene compartment that is to be undergoing transcription. And this enzyme is what we call the cyclin-dependent kinase. This is the checkpoint enzyme which ensures that we are not sending any cell with an aberration into the system or into the human body. Now, paying attention to the right explanation as to how Apml develops. 1. We know before any protein is produced, there should be a transcription. And after transcription, the messenger RNA is traveling into the cytoplasm where protein synthesis actually occurs within the ribosomal compartment. So, we will talk about ligands, enzymes, and the different steps that these cells go through. But the best thing we should know is that, are these protein, ligands, hormones that we are going to discuss falling under repressors, suppressors, or activators? When we talk about repressors in genetics here, pertaining to this explanation, we mean that any substance that has the propensity to resist transcription, that is the formation of the messenger RNA, or inhibit the transcribed message, which is the messenger RNA, from undergoing translation, is what we call a repressor. So repressors make sure that the transcription and translation does not progress, which means gene expression is inhibited. Now, suppressors make sure that the cell that is developed from the transcription is not progressing through the cell cycle. So in that stage, it halts the process of differentiation, looks into it if there is any DNA damage where it repairs, so there is DNA repair in there. And lastly, it switches on the apoptotic process, so that if that cell is aberrated, it channels it through the programmed cell death. So we've talked about the repressors, we talked about the suppressors, and now what are our activators? So the activators are those enzymes that the ligands or transcription factors attract. And when they attract them, they join them into a dimer and begins a cell cycle progression. And in that stage, there is phosphorylation of the specific genetic area on the chromosomes, and they are transcribed. Also, when there is activators present, there is cell cycle progression and there is more DNA synthesis. In this way, there is cell progression and the cells proliferate. So if it is about production of red cells, more red cells are produced, and if it is about leukocytes, more leukocytes are produced. And this is what we call activators, and activators help in proliferation of a particular lineage of cells. And a typical example of these activators is the histone acetyltransferase that activates the cyclin-dependent kinase enzyme that causes proliferation of cells and DNA synthesis and all that, just like we have mentioned previously. This means that our discussion about Apml will definitely lie on what factors are activating the cell progression and what factors are inhibiting the proliferation of cells and the cell cycle progression. So we will talk about retinoic acid receptors. So retinoic acid is derivative of vitamin A. And when we say retinoic acid receptor, likely means that these are receptors that responds to the binding of derivatives of vitamin A. So we have two main that are relevant in the discussion concerning Apml. And when I say Apml, I mean acute promyelocytic leukemia. So the two receptors are the retinoic acid receptor alpha and retinoic X receptor acid. We have the RAR alpha and then the RXRA. So these two are nuclear receptors. When we say nuclear receptors, we also group them as transcription factors. Why? Because they have affinity to bind to specific portion of a chromosome to activate transcription that will produce a particular type of cell. The affinity is for a specific promoter sequence within the gene of interest, and that specific sequence of genes where these transcription factors can bind is what we call the retinoic acid response element. So it's just a sequence of genes where they have affinity binding to phosphorylate and to cause transcription of that gene of interest. So remember, we've mentioned RAR alpha, we've mentioned RXRA, and now we are talking about RARE, that is the retinoic acid response element. These are promoter region on our gene of interest. Don't forget that. Now, the two transcription factors, which are retinoic acids receptors, have the ability to form a heterodimer. The reason we say heterodimer because these two receptors are structurally different, because RAR alpha is not the same as RXRA. So when these two join at their C terminus, they form a dimer, and this is called the dimerization. So in the form of dimerization, they have affinity for their promoter regions. They bind to their nuclear component or the sequence of genes within the gene of interest, and they activate transcription. But what goes wrong that leads into Apml? Now we are into the genetic aberration. There is a translocation that occurs on chromosome 15 and 17. So we say it's a T15-17. And when this translocation occurs, it leads into an oncogenetic fusion protein because there is a promyelocytic leukemia gene that is forming a dimer with our retinoic acid receptor alpha, meaning it is no more forming the dimer with the RXRA, but it's rather forming a joint gene with Apml. And in this stage, remember that already the RAR alpha has affinity for the promoter region on the gene of interest. When this genetic aberration occurs, instead of attracting activators to progress the cell cycle through proliferation and DNA synthesis and production of more of a particular good cell where there is checkpoint availability, it rather attracts co-repressors, and the co-repressors that are attracted include the histone diacetylase, the silencing mediator thyroid receptor, and the nuclear receptor co-repressor 1. And these make sure that the cell hijacks the G1 stage of the cell cycle. 2, it activates the tumor suppressors, and these make sure that it repairs if there is any genetic aberration. And the last thing that the fusion protein does is to inhibit the cyclin-dependent kinase. This means there is an activation of cyclin-dependent kinase inhibitors, meaning all the function that the activators through cyclin-dependent kinase will perform, like cell proliferation, differentiation, and cell cycle progression, will not happen anymore. And these are the stages by which the cell is stopped from going beyond the promyelocytic stage, and therefore all cells accumulate between blastic stage and that of promyelocytic stage. And in this circumstance, there will be accumulation of more promyelocytes, and promyelocytes have got azerophilic granules. These azerophilic granules within promyelocyte fuse together and crystallize into a long strand that stains like a pink-purple color, and this is what we call the R rods. And when R rods form bundles of cells together, this is what we call fagot cells. So fagot cells, R rods in there showing azerophilic granules with immature nuclear component is a typical appearance of the film comment for an Apml patient. Don't forget that the oncogenetic fusion protein, which is the Apml retinoic acid receptor alpha, has got the retinoic acid still available in there and has affinity for all trans retinoic acid, that's ATRA. Even as this fusion protein accept co-repressors to bind together to hijack the cell cycle, when ATRA, which is a well-refined and the most potent retinoic acid, which is the vitamin A, binds to this oncogenetic protein, it causes the dissociation of co-repressors, which means they are no more going to inhibit the cell cycle because of the dissociation of the co-repressors, and therefore ATRA, which is the all-trans retinoic acid, is a good remedy for treating people with Apml. Now, the bone marrow will be occupied mostly by these immature cells, including blasts and promyelocytes. There is not much space for production of normal leukocytes, which is these white cells involved in fighting infection, and therefore the patient will be prone to infection. And as well, very low level of platelets and red cells may be produced, and the patient can suffer from weakness due to anemia and also be at risk of bruises or bleeding. These are some of the basic symptoms that would be seen, and just like we have talked about the cellular appearance, how it will appear on your blood film, like this picture is a typical example of a acute promyelocytic leukemia. Research makes it clear that the oncogenetic protein, due to translocation of chromosome 15 and 17, which is the PML RAR alpha, does not relate to Apml in isolation. However, there are other genetic aberrations that also contribute to this disease cause or progression. There is one receptor called the FLT3. This is the FMS-related tyrosine kinase. This is a receptor that interacts with the purine-rich nucleic acid binding protein, the PU1. This is the master hematopoietic transcription factor, and this relates with all receptors required within the myeloid cell progenitor progression. So the myeloid cells have got transcription receptors such as the granulocyte monocyte colony stimulating factor receptors, the monocyte colony stimulating factor receptors, and other receptors that switches the hematopoietic pathway onto a particular lineage of cells. So this PU1 hormone interacts with the FLT3 receptor and tells which particular cell is being produced or which receptor to be activated. But something fishy happens in the FLT3, which is a genetic aberration due to internal tandem duplication. So we call it the FLT3-ITD. This internal duplication ensures that there is constitutive autophosphorylation of the FLT3 even without ligand binding. I mean, even without a hormone binding to this receptor, it autophosphorylates itself, making the docking site active for binding of these transcription factors like the PU1. And therefore, the progenitor cells in the myeloid lineage keep proliferating, meaning that transcription is going on, it is producing until it gets to the promyelocytic stage, where because of the oncogene PML RAR alpha, it is not progressing further, and therefore blasts and promyelocytes keep accumulating, not progressing beyond that. However, there is aggressive production because it is being forced to produce more of that. This makes this combined oncogene of FLT3-ITD and that of the PML RAR alpha a very aggressive type of acute promyelocytic leukemia. And not leaving this behind, the NPM1, which is the nucleophosphorin 1, is also involved in the acute promyelocytic leukemia. People who are also on treatment for different cancers, who are on radiotherapy, are also said to be at risk of developing the therapy-related acute promyelocytic leukemia. And I believe you love this kind of education. If you're interested to see more of this, you can click on the like button, share it, and subscribe to this channel. And there are more of this kind of education that is coming, and I believe that this is a good source of reference to help us understand some basic stuff in our jobs. Thank you very much for your time.